What's going on, everyone? Today we will be wrapping up our cells lecture, and we're going to be talking about how cells divide and multiply, and then the different types of tissues within our bodies. We can see that during cellular division that we're going to have certain phases, and we'll go over each of these, but here they are listed. Interphase, which is the phase before mitosis begins, and then the 4 stages of mitosis: prophase, metaphase, anaphase, and then telophase. Before a cell divides, it's going to have to go through a preparation in order to do so, and that is going to be called interphase. So this is the phase that takes place before mitosis, and there is a lot of metabolic activity because we are duplicating organelles. We're going to be replicating DNA, making strands for both daughter cells, and then we're going to increase the volume and the size of these cells in order to then split the cell apart. And so that interphase is where the cell spends most of its time during cell division. The first stage of interphase is called G1. This is when the cell is metabolically active, breaking down a lot of energy and using that energy for reactions that are occurring. During this stage, we're going to be replicating most organelles because both of those cells are going to need to have all of its organelles. So by duplicating, we're going to make sure that the original cell, the mother cell, has those organelles, and then when it splits in two, that both the cells have those organelles. And then the DNA is not going to actually replicate until the next phase, which is stage S. And so this is the phase where DNA replication does occur. I'm not going to go too far into detail with this. You probably talk more about this in biochemistry courses, maybe other biology courses, but I will explain it a little bit here. So we have the original strand of DNA, and what's going to happen is that a protein called helicase is actually going to unzip and separate these bonds between the nucleotides. And so then what's going to happen is you have 2 strands. One strand is typically called the leading strand, and then the second strand is called the lagging strand. And what's going to happen is that there's going to be another protein, DNA polymerase, which comes in, and for the leading strand adds nucleotides going from what's called the 5' end to the 3' end. And so it's going to add those nucleotides as we go. However, on the lagging strand, it's going in the opposite direction, and so these nucleotides are actually going to be added in fragments. But basically, by the end of this DNA replication, what we're going to have is 2 strands of DNA. One's going to be, for each double helix here there's going to be one for each new cell. And the last stage of interphase is called G2. We're going to continue the growth of the cell volume. This way we can have enough size for 2 cells. It's an important component needed for cellular division because we're going to get a lot of those important proteins and important enzymes that are required for cellular function. Here we see an overview of mitosis, which is going to be the actual separation of this one cell into 2 cells, and we just talked about interphase, but there are going to be a lot of stages in between this cell becoming just one big cell with multiple organelles and 2 sets of DNA for it to actually divide apart and then become these 2 identical cells. The first phase of mitosis is called prophase. And before I start explaining what's going on at each phase, I do want to point out that there is a lot going on at the molecular level, and it gets a lot more complex than what I am about to explain to you guys. But I'm just giving you guys a general overview and, what you guys need to know for this course. So again, this first phase is called prophase. During this phase, what we're actually going to see is that the nucleus of the cell is going to break apart, and then we're going to get a condensing of chromatins into chromosomes, and then the centromeres that you see on both sides of the cell, we're going to have microtubules span out from these centromeres and basically form these mitotic spindles and bind to the chromosomes. And eventually what we're going to see in the next phases is that these chromosomes are going to be split apart. So each new cell is going to have its own set of DNA. During metaphase, what we're going to have are these chromosomes are going to start to line up in the center of the cell. And what we're going to have these microtubules basically pushing and pulling to line up these chromosomes in that middle. And so, they all line up at what's called the metaphase plate, and those fibers eventually in the next phase are going to split these chromosomes apart. So we get one set of DNA for the first cell and then one set of DNA for the next cell. And we see that here during anaphase. So during anaphase, the centromeres will split and the chromosomes will be ripped, pulling them towards opposite poles of the cell. Sister chromatids are now considered individual chromosomes. And so, what's going to happen if you don't have this phase? Basically, you wouldn't be able to separate the sister chromatin, and then all would wind up in one cell. And that's not good because both new cells are going to need to have this DNA in order to form more proteins. And so during anaphase, we're just separating those chromosomes, and then we're pulling them towards the opposite side of the cell. The final stage of mitosis is going to be called telophase, and during this phase, basically, what we're going to have is a reformation of that nucleus around the two sets of DNA around all the chromosomes for each cell. And, once we repair the nuclei, you're also going to see that these cells, again, are going to divide, and they're going to split apart into two new cells that will then go into interphase. There are many different types of cells in our bodies, and they're going to vary in size, shape, and function. Some of them do not even possess DNA. Some of them are not going to replicate. Some of them are going to be involved in your nervous system. Some of them will be involved in, the respiratory system. And so we have a lot of different cells, each which have their own specific functions. These cells will come together to form different types of tissues, and we are going to discuss the primary tissues in our bodies, which are epithelial, connective, muscle, and nervous. The first tissue we'll discuss is epithelial tissue, and you probably spent hours in anatomy trying to memorize simple squamous, cuboidal, columnar, and all the different places that these tissues are found. But just a general overview, that epithelial tissue covers our body surfaces, so you'll see it in the skin. It also lines hollow organs and ducts. You can find epithelial tissue lining our lungs, our kidneys, blood vessels, and they also will be used to form glands. Connective tissue is the most abundant in our bodies, and within connective tissue, you're going to see fibroblasts and collagen, and it's used to help bind tissues together. You see it in between tissue types within our bodies. It's going to help support and strengthen, but you find it in a lot of different areas within our bodies, such as the subcutaneous layer underneath our skin. Adipocytes are considered connective tissue, even though they don't have fibroblasts. The connective tissue can also be found in our muscular system. Tendons are made up of this collagen fiber and fibroblasts, and even though they don't contract, they help to be a part of our skeletal muscle system. It's also found in certain structures of the body, including the trachea and the larynx, within our bones, and even the blood. The blood cells are considered connective tissue. Muscle tissue comes in three different types. These muscle fibers are going to contract in order to move. Now, the three different types are skeletal muscle, and so when the skeletal muscle tissue contracts, we're going to be moving the skeleton. When smooth muscle contracts, it's typically going to be transporting something or playing a role in transporting something, such as blood, such as air, such as nutrients. And then cardiac muscle is the muscle tissue that makes up our heart, and so this is going to produce tension and force in order to eject blood out into our bodies. Last, we have nervous tissue, which makes up our nervous system, and so it's going to sense changes with receptors in the body's external and internal environment, and we have neurons, which are going to generate action potentials in order to communicate things across the body. Sometimes it's sensory inputs, sometimes it's motor outputs, sometimes it's just communication between neurons, and these neurons are constantly firing using electrical chemical gradients in order to signal back and forth. And then we have neuroglia, which helps support the nervous system. To conclude the cells chapter, we are going to talk about cellular junctions. Obviously, multiple cells are going to come together to form tissues and organs. So these cell junctions help keep these cells together, and we'll talk about all the different types of these cell junctions. First, we have what's called a tight junction, and you see this in the lining of your stomach, and it's very important because if it didn't exist, then the contents of the stomach may leak out into the bloodstream, which would be very negative since you have things like acids within your stomach in order to break down certain foods. And so tight junctions consist of web-like strands of transmembrane proteins that fuse together the outer surfaces of adjacent plasma membranes to seal off passages between the adjacent cells. And so having these tight junctions are going to prohibit things like, contents of the stomach from leaking out into the body. Next, adherins are going to contain what's called plaque, which contains these actin microfilaments and also these transmembrane glycoproteins, which are going to help tether these two plasma membranes together. We also have what's called desmosomes, and how adherins had these actin filaments to help bind the two cell membranes together. Desmosomes are also going to contain plaque, but they are going to have intermediate filaments instead of actin filaments. Hemidesmosomes are a lot like regular desmosomes, but instead of connecting two adjacent cells together, it's actually just going to connect a cell to a basement membrane. Gap junctions are going to have proteins called connexins, which form these little tunnels called connexons, and in between these tunnels allow for the passage of ions and secondary messengers from one cell to the next. So you could see that a neighboring cell can pass on these ions and secondary messengers to signal events in a neighboring cell. That concludes our discussion on cells. Thank you guys very much for checking out these videos. In the next few videos, we are going to be discussing metabolism and talking about how energy is transferred within our cells and all the chemical reactions that take place. Thank you guys so much, and I will see you in the next video.